We're talking about the genital system development, and I'm Sananur Rahman. Now we're talking about the definitive stage, which is the second part of this lecture. This definitive stage is further divided into an overview in which we'll discuss what the definitive stage actually is. Then we'll see how the testes is formed and how an ovary is formed. We'll see the basic differences in the development of the testes and the ovaries, and we'll also take a look at what similarities lie in the process. So starting with the definitive stage, so now the indifferent gonad, it has to develop into an adult gonad, developing depending upon the chromosomal complement of the individual. It can either develop into a testes if it has an XY complement, or it can develop into an ovary if it has an XX component. So how does this development takes place? So first of all, let's take up the development of testes. So the primitive sex cords, these were the primitive sex cords in blue, in pink. These are the primitive sex cords which are actually formed by the proliferation of the surface ectoderm, the surface ectoderm being the coelomic ectoderm or the coelomic epithelium. So the primitive sex cord, they increase in length, they continue to increase in length, hence they penetrate this this reproductive ridge further and extend deep into the central part of the indifferent gonad. These sex cords, these are extending deep into the medulla of the indifferent gonad. At this point in time, they are called the sex cords. They are called the testes cords or the medullary cords. So what happens from here on outwards? Now towards the hilum, so this area is the hilum. These sex cords, these regress. So here you can see there's a regression of the sex cords towards the hilum. Now the tiny cell strands which are actually regressed, which are actually broken off from these sex cords, now they come to lie together with each other. They anastomose with each other. First of all, they undergo regression, then they anastomose with each other. As a result of the anastomosis, the sex cords become horseshoe shaped. Here you can see they become a horseshoe shaped structure. So now they get canalized to form the seminiferous tubules. So moving on, these sex cords, now they are being canalized. This is the canalization of the sex cords, and canalization of the sex cord result in the formation of seminiferous tubules. So in between these this formation of a horseshoe and the formation and the canalization, the sex cords, they attain communication with these rete testes, and this is how a communication is being formed from the distal proximal end to the distal end of the of the developing gonad. So anastomosing tiny cells get canalized to form the rete testes. So here you can see these are the anastomosing tiny cells which anastomose with these medullary sex cords. As a result of these anastomosis, rete testes, that is the communication between the two, has already been formed. So the communication is established between the seminiferous tubules and the rete testes. So recapping it once more time, keep an eye on the figure, you will be able to understand it easily. So first of all, these seminiferous tubules, at this point in time, these are the primitive sex cords. These primitive sex cords, which are actually the proliferations of the surface coelomic epithelium, they proliferate deeper into the hilum of the this developing gonad. These cells, that once they get into the towards the hilum, these cords, they now regress. The regression of the cord causes tiny cells of which have been released from the cord to come together with each other. They come together with each other and form a horseshoe-shaped structure. This horseshoe-shaped structure has been formed. Now leave the horseshoe-shaped structure here. Now focus on the seminiferous tubules. Now the seminiferous tubules, they begin to canalize. They form a canal within themselves. These canalized primitive sex cords, which were now called the testes cords, now become the seminiferous tubules. Now there's a small anastomosis occurs between the cells of the rete testes, which were the cells which were the horseshoe-shaped cells, and the seminiferous tubules. The communication between this rete testes and the seminiferous tubules helps in the connection between the proximal and the distal end of the developing gonad. Slightly difficult, slightly confusing, but if you keep an eye on the figure, you'll be able to understand it completely. So now let's talk about the cells of the seminiferous tubules. Now cells of the seminiferous tubules are actually of two types. We have the sex cord cells, which are the cells that are actually derived from the coelomic epithelium. They're called the sustentacular cells of Sertoli. So the Sertoli cells or sustentacular cells of Sertoli are actually those cells which were actually derived from the coelomic epithelium. The second kind of cells are the primordial germ cells, which migrated from the wall of the yolk sac. These are the primordial germ cells. They are responsible for the formation of spermatogonia. So, moving on with the development of the testes, the mesenchyme, it migrates beneath the coelomic epithelium. So this in brown is the coelomic epithelium. Now what happens? The mesenchyme, it migrates below the coelomic epithelium. It forms a tough fibrous layer. This in green you can see in this figure. It forms a tough fibrous layer which surrounds the developing testes. This is known as the tunica albuginea. So tunica albuginea is a layer of mesenchyme that actually comes to lie beneath the coelomic epithelium and covers the developing testes. Here you can see this in green is the tunica albuginea. So it separates the sex cords. Now the sex cords, which are now called the seminiferous tubules, from the coelomic epithelium, which is actually the germinal epithelium. So it causes a separation between the sex cords, which are now called the seminiferous tubules, and the coelomic epithelium. Here you can see this layer in green is separating the layer in brown and the layer in blue and pink. So blocking its this blocks the contribution of the germinal epithelium or the coelomic epithelium to the formation of sex cords permanently. So the sex cords that have been formed will stay there, and no new cells would be contributed from the coelomic epithelium towards the development of the sex cords. So now the mesoderm also forms mediastinum testes. So fibrous septa arise from the mediastinum. So fibrous septa, here you can see they are arising from this tunica albuginea. They extend towards the periphery. So they are extending towards the periphery, divide the testes into various lobules. So the mediastinum is also the mesoderm is also responsible to form the mediastinum testes. And how is the mediastinum testes formed? By the fibrous septa, which arise from the mediastinum and extend towards the periphery. They divide these fibrous septa. They divide the testes into various lobules. Now the mesoderm around the seminiferous tubules, it forms the interstitial cells of the Leydig. So now interstitial cells of Leydig are formed, and how are they formed? From the mesoderm, which surrounds the seminiferous tubules. So it's a lot going on. So I'll recap it. What happens? Focus on the figure. So what happens is, first of all, we had this coelomic epithelium. The coelomic epithelium in brown. This in brown is coelomic epithelium. This coelomic epithelium undergoes proliferation. That is, it forms many cells. When a lot many cells are formed, these cells form finger-like projections, which are called the primordial sex cords, and these primordial sex cords now penetrate the developing gonad. So now these primordial sex cord, they elongate, and now they are called the testes cords or the medullary cords. They elongate and reach the hilum. Once they reach the hilum, these testes cords or the medullary cords, they begin to break, and the cells that break off actually clump together and anastomose to form a horseshoe shaped structure. At the same time, the remaining the testes cords or the medullary cords that have not broken down, they undergo canalization to form the seminiferous tubules. Now there's a development of connection between these horseshoe-shaped cells and these seminiferous tubules, and hence there is a continuation from the proximal to the distal end of the developing gonad. At the same time, the mesenchyme now comes to lie beneath the coelomic epithelium. Now, when the mesenchyme comes to lie beneath the coelomic epithelium, it forms a tough layer of a tunica albuginea, which separates this coelomic epithelium from the developing seminiferous tubules. So now this coelomic epithelium will not contribute any more cells to the developing sex cords. At the same time, various fibrous septa are formed in the mediastinum of the testes, and they extend towards the periphery. These septa now divide this testes into various lobules. Now the mesoderm around the seminiferous tubules, it forms the interstitial cells of Leydig. So this is what is actually happening and how the development of testes takes place. So now the tubules of the rete testes. So these cells were the cells of the rete testes. So now the tubules of the rete testes, they extend into the mesonephric tissue. Here you can see these tubules are extending into the mesonephric tissue. They now join the mesonephric tubules. These are the mesonephric tubules. These are being joined by the tubules of the rete testes. They become the efferent ductules of the testes. So the efferent ductules of the testes are formed by the tubules of the rete testes, which were extending into the tubules of the mesonephric duct, and when both of them anastomose with each other, they result in the formation of efferent ductules of the testes. So now the duct of epididymis, this is the duct of epididymis. The vas deferens, now the vas deferens and the seminal vesicles and the ejaculatory duct, they are formed from the mesonephric duct. So the mesonephric duct, it gives rise to the duct of epididymis, the epididymis itself, then the vas deferens, the seminal vesicles, and the ejaculatory duct. So now the testes develop, they are projected into the coelomic cavity and are suspended from the posterior abdominal wall by a mesentery, which is known as mesorrhecium. So testes, it is being suspended in the coelomic cavity from the posterior abdominal wall by a mesentery, which is known as mesorrhecium. So moving on, we have now the development of ovary. So initially we were talking about the development of testes. In development of testes, we saw how various processes took place to form the adult testes in an XY complement individual or a zygote. Now the zygote has an XX complement. So let's see what happens. So the primitive sex cord containing the primordial germ cell, they extend into the medulla. So this was the stage of indifference. In this stage, these are the sex cord, these are extending within the medullary cavity. And these sex cords, they also contain the primordial germ cells. Now they dissociate into irregular clusters of cells. So this is the dissociation of the medullary cords into irregular clusters of cells. Now, these irregular clusters of cells are called rete ovary. So, we had rete testes in males and we have rete ovary in females. So, they occupy the medullary part of the ovary close to the mesonephric tubules. So, it is almost the same part as that of the rete testes. They occupy the inner portion or the medullary part of ovary, which lies close to the mesonephric tubules. So later they disappear, the rete ovary they disappear and are replaced by vascular stroma, which forms the ovarian medulla. So here is what is different from the development of the male counterpart. Now these rete ovary they later disappear and are replaced by vascular stroma, which is responsible for the formation of ovarian medulla. Here in the figure you can see this is the coelomic epithelium from which the primitive sex cords are extending within the lumen. Now here you can see these sex cords, and these are the irregular cells, the rete ovary, which have been formed. These rete ovary will finally be degenerated and be replaced by vascular stroma, which will be forming the ovarian medulla. So the surface epithelium of the female gonad, it continues to proliferate. So this surface epithelium, this coelomic epithelium, it continues to proliferate. In the 7th week of development, it forms a second generation of sex cords. So a second generation of sex cords is now being formed in this in the developing female gonad, which was not formed in the developing male gonad. So in the 3rd month, the cortical cords get fragmented and form isolated cell clusters. So here you can see this. These are the cords that have been fragmented and now they have formed isolated cell clusters. So the primordial germ cells are formed. So primordial germ cells now have an oogonia and coelomic epithelial follicular cells. So what happens? Let's go back to the figure, the previous figure. So here, this is the coelomic epithelium. These are the primary sex cords. Primary sex cords are now migrating into the lumen of the developing gonad. These primary sex cords, they form the rete ovary. Rete ovary then undergo they are reabsorbed because they disappear and are replaced by the vascular stroma, which will form the ovarian medulla. At the same time, this surface epithelium, it undergoes proliferation, forms a second set of the cortical cords or the sex cords. These sex cords then undergo these sex cords they get fragmented as well. They are fragmented and form isolated cell clusters. So these are the cell clusters that are being formed. Each cell cluster has a central primordial follicle, has a central primary germ cell. This in red is the germ cell, and these surrounding cells are the cells that are derived from the coelomic epithelium. So primordial germ cells form oogonia and coelomic epithelial cells form the follicular cells. So this central red cell is known as the oogonia, and the surrounding cells are which are formed by the coelomic epithelial cells are called the follicular cells. Now they form the primordial follicles in the females. So primordial follicles are being formed in the females. So now let's see how the surface epithelium changes. So the surface epithelium of the ovary, it flattens. So now there's flattening of the surface epithelium. Here you can see the surface epithelium now flattens. It forms a single layer of the cells which is continuous with the peritoneum at hilum of the ovary. This is known as the germinal epithelium. So it forms a single layer of cells, and this is continuous with the hilum, with the peritoneum at the hilum, and it is called the germinal epithelium. The mesoderm forms thin connective tissue covering the ovary and connective tissue stroma of the ovary. So the mesoderm, it only forms a thin connective tissue that covers the entire ovary, and it is also responsible for the formation of the connective tissue of the ovary. However, in the male counterpart, we had tunica albuginea, which was formed by the mesoderm, as well as the septa in the mediastinum of the testes, which cause lobulations in the developing testes. So now the ovary is suspended from the posterior abdominal wall by a mesentery, which is called the mesoovarium. So mesoovarium is responsible for the suspension of ovary from the posterior abdominal wall. So again, recapping what actually happens and how the chromosomal complement results in the formation of adult phenotypic appearance of sexual organs. We have a primordial germ cell, which is 44 plus XY, that is the male counterpart. So now the male under the influence of SRY gene on Y chromosome, the indifferent gonad, it develops into testes. And what are the differences? The medullary cords, they develop as well as they persist. There are no cortical cords, so no cords in the cortex are formed, and the tunica albuginea develops. Now again, we have the primordial germ cell with a complement of 44 plus XX, that is 44 plus a female component. There is absence of the SRY gene, as a result of which the indifferent gonad differentiates into an ovary. Now the medullary cords they develop, but they degenerate. The cortical cords they develop, and finally tunica albuginea does not form. So these are the differences which actually take place due to the genetic complement of the fetus, which help in the determining of phenotypic sex of the adult fetus. So what are the differences in development of testes and ovaries? Again, it's a very important question. Commonly it occurs it is always there in your exam or in your viva. So for testes, there is formation of only one generation of sex cords, that is the medullary cords, that result in the production of seminiferous tubules and the rete testes. When we compare it to the ovary, there is formation of two generations of sex cords. The first generation, the medullary cords, they form the stroma of the ovarian medulla, and the second generation of sex cord, they are responsible for the formation of primordial follicles. Now in testes, we have formation of tunica albuginea, which separates the seminiferous tubules from the surface epithelium. And in ovaries, there is no such formation, so no formation of tunica albuginea, hence the ovarian follicles are not separated from the surface epithelium. So this was all about this section. In this section, we talked about the development of ovaries as well as testes. It was a very extensive section, which and the recap of which I did at every slide so that you can understand it completely. Try to go with the figure so that you understand it. For further such sections, keep watching sqadia.com.